Prototyping of synthetic G networks is greatly improved by the use of cell-free protein expression systems. Our protocol describes the fabrication process of a multilayer microfluidic flow reactor and shows how such a device can be used for prolonged cell-free expression of GFP. Cell-free protein expression in combination with microfluidic flow reactors provide a rapid prototyping platform for the design of synthetic biological devices.
This entire setup is highly versatile and can be adapted for the conduction of any biochemical or chemical reactions requiring high levels of control. Ensuring the stability of the cell-free reaction solution is critical and difficult to achieve. Be sure to focus on providing sufficient cooling and utilize the tubing specified in this protocol. Fabricating and preparing the experimental platform requires the connection of numerous individual components, a process which can be difficult to follow without visual aids.
To begin, follow along in the accompanying text protocol to fabricate the microfluidic device and to set up the pneumatic control and flow pressure regulation, which will be used for controlling the valves critical to this device. To set up the off-chip cooling, begin by coiling a length of PTFE tubing onto the cold face of a Peltier element and secure the coil with tape. Ensure that one end of the PTFE tubing is connected to the reservoirs of the flow layer pressure control system, and that the other end protrudes no more than 1 cm from the Peltier surface. Next, insert a 5 to 10 cm length of PEEK tubing into the protruding end of the PTFE tubing. Apply a sufficient amount of thermal compound onto the hot face of the Peltier element and place it onto the cold plate of the water block. Ensure